how do things cross the cell membrane? And there's a lot of different ways that can happen. Sometimes it happens passively. It doesn't require any energy. And we'll talk about 3 examples of passive permeability. Starting with diffusion. Imagine that I show up with a big old box of pepperoni pizza. And I open the lid, and all of a sudden these pizza odor molecules leave the box where they are highly concentrated, and then they diffuse out through the classroom. Eventually, everywhere in the classroom there'll be a little bit of a pizza smell, because the odor molecules in that case have gone from a region of high concentration to low concentration. It's the same thing when you take a drop of dye or a sugar or salt cube and put it in water, and everything evens out. In the body, what we're saying is that if the inside of your cell has a low concentration of a solute, like salt, let's say, and outside the cell has a high concentration, these ions will cross into your cell on their own, no energy required, until things are evened out. The explanation for that is chemistry and is physics and is for another class. Sometimes stuff can't just cross the cell membrane. It needs a helper. Facilitated diffusion is like diffusion because things are going from high concentration to low concentration, right? The pizza box still opens and all these molecules want to move, but sometimes they need a helper. Maybe a channel, maybe a carrier. These are just different types of proteins that are going to help move things across the cell membrane. The final passive process that's important in cells and cell membranes is osmosis. Water molecules can diffuse also. They also want to move from areas of high concentration to low concentration. If we see a U-shaped flask or beaker or something with a semi-permeable membrane in the middle, on one side we could see there's a lot of water molecules because there's not much solute. On the other side there's less water because there's so much solute, so many of these purple dots. So water is going to want to move across that membrane to even out its concentration. And indeed, it will do that, causing a rise in the volume of fluid on one side of this beaker. If that seems a little strange, let's look at an example inside the body. If you have a cell that has a high concentration of a solute, let's say a lot of blue dots, and then the outside environment has a low concentration of that solute, water will move in and out of the cell all the time, but more water is going to move into this cell and cause it to swell, maybe to burst. The opposite would be true if you drop this cell in, let's say you dropped it in a very salty environment. There's a little bit of salt inside this cell, but if you put it in a very salty environment, water is going to want to exit that cell to even out the concentration of water, causing the cell to shrivel. Normally, some water is coming in and some is going out, and everything is balanced. And at the risk of getting too dark, if we really want to take this example to an applied human life level, we can talk about Matthew Carrington and Jessica Strange, both Californians, both in their 20s, and both died from water intoxication. Because if you introduce a bunch of water to the body, that water is going to want to move through osmosis. In the case of Matthew, through a hazing at a college, and in the case of Jessica, through some dumb radio program where you were trying to win a prize. So in both cases, they took on a lot of water. That water tried to even out its concentration by moving into their cells. Some places in your body can swell. If that happens, the cells will swell. If that happens in the cells of your brain, though, they don't have much room to swell. So it puts pressure on the brain. You go into a coma and die from that pressure. So this is important stuff. This is, especially for people who are in nursing, even dentistry or sonography. I mean, when those people walk into your room, those patients become your responsibility, right? So you want to understand some of this stuff. Okay, let's finish with the active processes by which things cross the cell membrane. All of these require energy. Active transport is when there are proteins that are helping things move across the cell membrane, but now it's using ATP to actually power something and move these solutes against their concentration gradient, right? Because if there was a bunch of salt out here, it would move on its own into the cell to even things out. But let's say you don't want things evened out. Let's say you want a bunch of salt inside the cell. The cell is greedy, it's going to pull in as much salt as it can. Well, you don't want that salt to leave again. So you got to keep pumping it back into the cell. So you're actively using energy to pull stuff where it doesn't want to go against its concentration gradient. That's active transport. Next is endocytosis. Endo means within. Cyto is cell. So you're bringing material from outside the cell into the cell. And there are two kinds. Phagocytosis is cellular eating. And pinocytosis is cellular drinking. Pinot is also a wine. I'm not really into that so much. It's Californian thing to go wine tasting, not so much in New Hampshire. I went. I liked it, but I couldn't taste all the things, But I know that Pinot is a wine. So that will help you remember pinocytosis is cell drinking. What we're saying is the cell can envelop food particles, bacteria that are invading your body, or just large amounts of fluid and bring them into the cell. So we can see that in action here with this amoeba. Oh no. About to eat. So this is not happening inside humans, but here we can see one organism, this amoeba, slowly enveloping these paramecia, and it will bring them into its cell. So this is a type of endocytosis. Oh, now they know. Oh no. And that's it. And they turn into a blob of goo. So an example of endocytosis, but more specifically, phagocytosis, cellular eating. If the amoeba had just enveloped a bunch of fluid to hydrate the cell, that would be pinocytosis. The opposite of endocytosis is exocytosis, in which the cell moves material from its inside to the outside. And typically what that means is you have a vesicle, which we'll talk about later on, but a sphere or a packet of material that's going to fuse with the cell membrane to expel its contents. We can see a paramecium go through this process by watching the lower left corner. A little bit of time get you excited, but That's all it is. Just content leaving the cell is exocytosis. Okay, so for last little activity, here are some images that I put in to test your understanding of the terms that describe how things cross the plasma membrane. So I'll just mention a couple of these and then leave it to you. Hopefully you remember the pizza smell from earlier. What if I wanted to take away that pizza smell, right? All the odor molecules have drifted throughout the room. Now I want to somehow collect them and force them back into the pizza box. I don't know how you would do that, but for sure it would require some kind of energy. So that imaginary scenario is a form of active transport in which here the woman is sad because I took away the pizza smell, I pumped it or forced it back into the pizza box through active transport. And another example of that might be, let's say if you're trying to fill up your closet with tons of stuff. It wants to spill out onto the floor and just even out the concentration of junk, but you're trying to force more and more and more things either into or out of the cell. That would be another form of active transport. So take a look at these other pictures, see if you can figure out what's what, and then have a look at the key, which I will end with today. See ya!